Hello and welcome to the Bioinformatics Chat. Before we start, let me remind you about the Integrative Biology and Medicine Conference. Among other speakers, you will hear from Svetlana Kurina, University of Manchester, about the role of microRNAs in wound healing. You will also hear from Bartek Olczyński, University of Warsaw, about integrating gene expression data with chromatin structure data for better models of gene regulation. The conference will be in early October in Kiev, Ukraine. I'm here with Valentine Svensson. Valentine wrote a tool called SpatialDE and the paper SpatialDE Identification of Spatially Variable Genes. Hi, Valentine.
Hi.
So we will talk about your tool, but before that, tell us about yourself, about your background, education, prior experience, etc.
Well, I did an undergrad in pure maths in Lund in Sweden. And then I worked for a couple of years at a genomics facility in Stockholm that made me interested in biology. So I applied for a PhD position at EMBL, and I went there and I joined Sarah Teichmann while she was at the EMBL EBI, European Bioinformatics Institute. And I've been doing a PhD with her for the last 4 years, and I actually defended my PhD about a month ago.
Oh, congratulations.
Thank you.
So now your PhD is finished, and what's your sort of current employment or?
Well, I have some funding to stay here for a few months more, but then I'm going to go to Caltech in California for a postdoc.
Oh, pretty cool. So you already got accepted there?
Yes.
Mm-hmm. And SpatialDE, was that part of your PhD? How does it fit into your general research?
Yeah, it fits in. So my research for my PhD has been about cell differentiation and how to study that using single-cell RNA sequencing. And we have been doing this by essentially using a kind of analysis which is quite common in the single-cell field called pseudotime analysis, where you take a snapshot of data and you kind of order this all of the cells in this snapshot to form a kind of temporal trajectory or pseudotemporal trajectory based on the gene expression values. What we wanted to see is what genes do something interesting over time. And we found that using Gaussian process regression was a very good way of finding genes which had some kind of smooth and interesting behavior over time, but we didn't have to specify what kind of patterns they should be. And if we fast forward a bit, a year ago, I think there was this paper published in Science where they had done not single-cell RNA sequencing, but RNA sequencing which was spatially resolved. And this was a technology we thought was really interesting because it could complement our single-cell studies and allow us to investigate gene expression in a tissue context for example, immune cells and how they are located within different tissues. And we were wondering, how would we analyze this kind of data? We did also do our own experiment as a pilot for this, which we haven't published yet, but it's something we were working on. And we wanted to, in general, just find, okay, we have these 20,000 genes which are expressed throughout the data. Which ones should we start looking at, which they didn't know of before, which are doing anything interesting over space? And we figured that we could actually apply the same concepts that we did for our time course analysis before. So we can find these genes where like the variation in gene expression is dependent on the spatial coordinates. So we made this method and tool which can identify genes which are doing something which is correlated with the tissue structure or the spatial coordinates in the tissue.
So you mentioned pseudotime. Do I understand correctly that pseudotime has nothing to do with like actual time, so that you're not talking about the actual time series data? Could you explain a bit more how that works?
Sure. So that depends a bit on the experiment. We've done it in a couple of different ways, but I published a paper in Cell Reports a couple of years ago where we investigated blood development in zebrafish. So the zebrafish is actually a really good model for blood development because all blood develops in one concentrated region in the kidney, as opposed to in mammals where it's developed in the bone marrow. So you can actually sample all the blood progenitors in a fish in a pretty efficient way. And what we did there is we took a single fish and we sorted out cells that we knew came from one blood lineage, the thrombocyte lineage, which is the equivalent of platelets. So this is just one time point, one fish. And using this kind of pseudo-time analysis, we could actually reconstruct the development from progenitors to mature thrombocytes. And we could then analyze gene expressions over this development. We found we found that translation genes kind of go down before transcription genes and things like this.
Mm-hmm. So for pseudotime analysis, did you also use Gaussian processes?
Yeah, so that's what we found we found it to be pretty good when you don't have any prior information about what kind of curves you're expecting over time. And we used something, first, in the beginning, we were just using Gaussian processes for analysis once we had the pseudotime to kind of see which genes are doing something interesting over time. But we also found that there's this really good model called the Gaussian process latent variable model, which just assumes that all of your genes follow, to some extent, some of these Gaussian processes. And then you can actually learn the time values using this. And yeah, it's something that's worked really well for us so far.
Returning to the spatial variation, how do you actually obtain the spatially resolved data? How does that work?
So there are different techniques for this. In the paper, we highlighted 3 techniques for doing this. One is genome-wide, and the other 2 require you to design probes for a set of target genes. So for the genome-wide technology called spatial transcriptomics, and this is the one that was published in Science last year, what you do is that you freeze your tissue and you slice it up with a microtome, and then you treat the slice in a particular way, and you put it on top of a kind of inverted microarray. So it's a microarray where the probes that would normally attach to target genes have poly-T DNA on them. And then on the other side of the probe that's connecting to the actual array, there's barcodes which are spatially resolved. So you know which DNA barcode corresponds to which coordinate on the array. And then you let this tissue permeabilize on top of this array, and RNA from the tissue will just float down to the array and attach to these coordinates. And then you kind of treat it as almost like a single cell RNA-seq protocol, but with more material, because the spatial coordinates on the array are on the level of between 10 and 100 cells rather than one cell. For the other technologies, they're more imaging based. So they're based on single molecule fluorescent in situ hybridization. So you have to design a number of probes for genes, and then you can image them. And the technologies which are more interesting for this kind of test that we're doing to find interesting genes, these would be technologies where you can do many probes. And there are a couple of techniques, one called seqfish and another called merfish. They're conceptually similar. I think merfish has some error correction and seqfish does things a bit differently, but in the end you make like probes which have multiple, you basically do multiple rounds of hybridizations of things which create different colors. And this way you can kind of design a coding scheme from that to which probe different DNA came from. And that way you can kind of, using a bunch of images, deconvolve this into which genes were spotted in the images, or which RNAs rather. These have been scaled up, so seqfish can do, or could do in the paper they published, 250 genes, and there's a paper for merfish where they did about 1,000 genes. I'm hoping this will scale up in the future because it's a really high throughput technology. So in one of the merfish papers, they did one experiment where they could analyze this like single cell expression level of 100 or so genes for over 35,000 cells in a tissue.
So what is the typical spatial scale of this data? Are we talking like on a cell size scale, or is it like 1 centimeter, 10 centimeters?
Yeah, that depends a bit on the technology. The spatial transcriptomics is less resolution. So as I said before, so the way that kind of array is designed, I think it's about 2 millimeters times 2 millimeters. Oh, maybe I should bring up the technical specs so I don't say something wrong. And there you have like this kind of regions on the array, which are circular, having a diameter of 100 microns with 100 micron space between them. So that's kind of the resolution.
With the array, the size of your sample of your tissue has to correspond to the size of the microarray itself, right? It has to be on the same scale.
Yeah, you kind of want to do something which is on that scale. It's going to be the best way. They did do a cool paper applying that to plants. I think it's in Nature Plants. They used it in different plants, but the plant regions that they used were kind of small, so they only got like 5 or 6 spatial coordinates, which in the end you couldn't apply my method to because it's kind of too little data. But I thought it was really cool anyway, and you get these really cool pictures of, buds from different plants and gene expression in them. On the other hand, the in situ hybridization methods, those are actually subcellular, and we were thinking about like whether we could also do something with that to see like whether some RNAs are localized at the sides of cells or something like this. But that's more of a different kind of analysis than we did here. But I think it's pretty interesting, like what you can do with the subcellular resolution of data.
Mm-hmm. But what about the upper bound of this? Like if you wanted to investigate some spatial patterns on the scale of like the whole organ, like liver, would that work?
Yeah, there's a limit to that as well, how large the images can be. Yeah, I can't remember off the top of my head. But in the latest Murphy's paper, they did make a point of making a very, very big image that by kind of joining together multiple images that were imaged after each other. But the areas are much smaller than the area for the spatial transcriptomics.
So is the data between different experiments comparable as well? So you can like make multiple snapshots, but then like how large would you expect the batch effects to be?
It depends on what thing you want to do. So with the in situ hybridization things, of course, you have like a limited number of genes that you look at. It could be the case that if you looked at the same genes as investigated by spatial transcriptomics, you might see something interesting there as well. Though the structures that you would see in the spatial transcriptomics data would be more macro, while the structures in the other data is more on the smaller scale. So in the paper, we looked at one seqfish dataset and a couple of spatial transcriptomics datasets. But one of the ST datasets that we looked at was from mouse brain, from the olfactory bulb, while the only available seqfish data is also from mouse brain, but the hippocampus. We did see some genes being common between these, and it's probably some genes which are not extremely functionally relevant for the tissues that were studied, but were common for just being in neurons or something like this. So that kind of suggests that maybe they could be combined in some way to work something out with the neurons. An issue, of course, here is that, this is if you just want to compare the samples and the individual, locations in these different technologies and just combine them on a gene expression level. But of course, I don't think there's a reasonable way to combine the spatial coordinates because like they're on different coordinate systems.
Mm-hmm. So like you expect it to be a problem to like fix the origin of every coordinate system of every experiment? Is that what you're saying? Yeah,
or how do you align a small piece of one tissue that's from one mouse and another small piece from another tissue, which is from a different mouse, mm-hmm. from a different continent? While the cells themselves might be comparable because one is one kind of neuron and the other is another kind of neuron, and you can compare how these different kinds of neurons are in the different experiments. But the spatial coordinates themselves probably will depend on, what kind of mouse did you use and the mouse itself.
Right.
I mean, of course, I know that this kind of image registration is something that's common in like medical imaging, but I'm not sure like how that applies to in situ hybridization.
what kind of file format do you use to represent the spatial information? So I guess your reads are barcoded, and then you have like a table that gives coordinates for every barcode. Is that it?
Yes, that's at least that was the data we got from the people at Spatial Transcriptomics. So we performed. So I've used when I've used the public data, actually together with the paper, there's a website which has pre-processed data, which is really handy actually, and I wish more papers did this. So there I could just get a table where the row names are the spatial coordinates and the column names are the different genes, and then in the matrix is just the gene expressions. So that's quite handy. When we got our own data back from them, we got basically reads and then a table that's translated a barcode into coordinates, and then we have to kind of join that together. It wasn't a lot of work actually, but yeah, that's the data format.
Mm-hmm. And what kind of data does SpatialDE expect? Does it support like one fixed format or multiple formats?
So SpatialDE expects two tables basically. One table which has names of samples in the rows and names of genes in the columns, and then it's just a gene expression matrix in general. And the other table it expects is a table which has the sample names in the rows again, but then also some kind of spatial coordinates in the columns, and you can specify the names of these columns for whatever you want. So actually I tried it on some other data where the coordinates were like names of people, and it's also something that works, but you have to change what you use for the covariance function.
Okay. So SpatialDE supports any number of dimensions, right? And I think you even mentioned in the papers that you can treat the time as one of the dimensions.
And as I mentioned in the beginning about the origins for this was how we have analyzed a lot of data before, is to use exactly the same model but over time. Then we used a package to do this called GPy, and we wanted to kind of accomplish a couple of things here. So first of all, we wanted to make it very specialized for these kind of spatial studies, but we also wanted it to be very, very fast. So when I've been using GPy for the other time series data before, it takes something like 100 CPU hours to do a genome-wide search, while it takes 8 minutes for this package. And something which I've been doing when I've been teaching about how to do time series analysis is looking at the data set, which I've also included in the paper, which is a really cool data set, which we didn't make, but it's a time course of frog development, like embryonic development of frogs. And they picked basically embryos at very, very close intervals. So it's a very high resolution time course. So it's really good to test these kind of models to find like temporal variation. And when I run that on a cluster, it's taken about 100 CPU hours. But here I'm reanalyzing the data in the paper, getting similar results, but much faster. But then it's kind of like saying that time is like a one-dimensional space. And in theory, you could kind of combine these things to have like a two-dimensional space, but also have a time course. I'm not sure like how you would assay this, but
that's what I was thinking about, right? So that's not currently supported.
It's definitely supported, so you can put in any number of dimensions as you want. I just don't think there exists any data for this.
But I was thinking that depending on the nature of each dimension, you probably should treat it differently. So, for example, if you have 3 spatial dimensions, they are more or less like not equivalent, but interchangeable. But the time is very different kind of dimension.
Yeah, it's true actually, because that's a good point. Because the way we do it now, we're calculating pairwise distances between points. And of course, in 2D space or in 3D space, pairwise distances make sense because the distance unit is the same in X coordinate and Y coordinate and Z coordinate. But if you have a time, if you measure time in seconds, it's going to be different from if you measure distance in centimeters. So it's a good point to think about how we should deal with that. To make some kind of like smoothing, or basically, I mean, the way you would do this in a more general package is that you would, so the way the model works is you calculate kind of distances between things, and then you're saying, I think things which are close at this distance covary, and that way you can kind of make predictions and inferences. But you can also say that, so the way I've done it here is that I assume that things will covary at the same scale and the x coordinate as the y coordinate. Mm-hmm. But you can also change that and say that actually I want to allow things to covary at different rates in different dimensions. And sometimes this makes sense even for spatial things. So I'm trying to think of a good physical example, but say that you're at a beach, for example, and you're seeing waves coming in towards the shore from the sea. Mm-hmm. You will see more variation, or you will see like more interesting patterns. I think at least that's how it looks in my head, of, waves being varying in a way that's kind of parallel to the actual beach, as
right.
opposed to varying in a orthogonal way to the beach. So then you would want to say that actually I think the length scale of covariance is shorter in the y coordinate than it is in the x coordinate. And similarly, if one was time and one was a spatial coordinate, you can say, well, I measured space in centimeters and I measured time in seconds. So the units in my table I have here are not really comparable. And therefore the length scale for seconds should be separated from the length scales in centimeters.
Right, and I imagine there are plenty of reasons for even the pure spatial data to be also for it to vary differently in different directions, because although in theory our physical world is, I think, isotropic is the correct term for that, but still we have, for example, lateral symmetry, right? So left, right will have maybe different meaning than forward versus back. And then there's, of course, gravity. So up, down will have some separate meaning from that.
Yeah. And we did, I did do a couple of experiments of trying the different things to see what we should support in the tool. And we found that looking at tissues, the kind of things that most people are interested in will be the ones which are varying in the same way in either direction. And that will also cover cases when only one dimension varies, but it will get a different p-value in the end than if you say that there's absolutely no covariating expression values in the x coordinate. Everything is in the y coordinate, which happens, of course, in some tissues. I mean, even one of the samples in the paper is this brain tissue I mentioned before. And if you allow the covariance to be separated, it has an easier time finding genes which are determining the kind of left-right symmetry between the lobes. But we kind of thought, but it's more general, being in the different directions actually covers most of the cases, and the slight improvement in the significance tests are not really worth it for the extra hassle of dealing with multiple length scales.
Right.
But when we have time courses, which might then be coming in the future, we have to rethink that, because as you say, the units will be different.
Mm-hmm. And I was thinking that even for purely spatial data, if you go a bit further, not just allow different measurements in different directions, but also I'm wondering if you could build into the tool some kind of PCA analysis so that it would figure out the correct directions by itself, so you wouldn't rely on the correct alignment of your whatever it's called, right, of your assay to the correct axis, but it would figure out the axis automatically. I guess you could do that separately from the Gaussian process, right? Just do a normal PCA on your data.
Yeah, I'm not sure. So I'm not sure if I completely follow, but I can give, I can relate it to something that sounds like it's relevant to what
Okay.
you're saying. So there's a couple of papers from a few years ago, both I think in Nature Biotechnology, one from John Marioni's group and one from Rahul Satia. And what they did is that they did single-cell RNA sequencing of cells without knowing the spatial coordinate. But they had a reference with like a few genes where they knew in an organism where those genes like usually were. So they could use this reference with 10 or so genes to map their single cells into likely spatial coordinates. And in such a way, like, taking these non-spatial samples and make spatial coordinates for them based on data from some markers. And I think that's kind of what you mean. So similarly to how people do pseudotime, you could think of like pseudospace.
Yeah, I see what you mean, and it's interesting. But I guess this method, it wouldn't have a great resolution, like a great precision.
No, so what we're taking advantage of here is that we know this extra information, and I should say like getting this extra spatial information is very expensive compared to not getting it. Like you have to deal with your samples in a very particular way, and it's quite costly as well to do it. So we want to make sure to use that information once we get it.
Mm-hmm. Yeah, I was thinking about the precision of this information, right? How, like you're saying that we can map in a precise way where the particular genes are expressed.
Okay, do you mean in the data we have, or the thing I explained about the backmapping?
Yeah, the thing you just described.
Oh, so the backmapping thing, yeah, I don't think there's been a comparison between having the actual coordinates. They did have interesting results in the papers they published. But I think, yeah, you gain a lot of confidence by actually looking at the location.
So you alluded a bit to how Gaussian processes work, but do you want to get into like a bit more detail? What is this model?
Yeah, sure. So the rough idea of it actually comes from, I think, about 100 years ago or so in South Africa, where there were gold miners who tried to figure out like what's the best location to mine for gold next. And imagine that you're doing this thing where you kind of dug a mine somewhere, and then you found a bit of gold, and then you dug a mine somewhere else, and you didn't find any gold. But then kind of a bit from both of those places, you dug a third mine, and there you found more gold again. So then you want to know, okay, where should I dig my next mine, where is the most likely that I will find gold? And the data you have, well, so you have this underlying assumption, which is that like things are in these veins under the ground. So you think that things should be like close to where you found gold before, you will find more gold. And the closer you are to a place where you already found gold, the more certain you are that you will find gold again. So you're basically saying that around an area where I found gold, I should find more gold. And around an area where I did not find gold before, I will not find gold. And then as you go between these things, you get more certain as you're moving towards the mine that had gold from the mine that did not have gold. And you can phrase this as a covariance, which depends on the coordinates of these mines. And this way you can say, okay, if I'm digging in between the two mines that had gold, I have a higher expectation of finding gold there. And then how quickly you think these like expectations decrease is something that you can estimate if you have a lot of data, much more than 3 points. And that's kind of the basis of it. So you're just saying that I don't really know how the gold vein looks like, but I expect it to have a higher density close to the places where I knew I could find gold. So the way we parameterize this is that we write a function saying how these measurements of, for example, gold or gene expression are covarying with other coordinates and depending on other coordinates rather than what is the actual values at these coordinates. So then you can be data driven. So you won't know anything if you don't have any data. You will just like say, oh, well, if things are close to each other, then they should have the same, but I don't know what is there. But if you then also supply some data, it will inform you about where it is certain that things will have high values and where it is certain things will have low values. So it's a model of the covariance structure. And together with the covariance structure and data, you can predict with uncertainty how things will look where you did not actually sample data.
Right. And just to connect your explanation, your golden explanation to the paper. So I think the covariance structure corresponds to kernels, right?
Yes.
And so the kernel you just described, it sort of just measures how, I'm wondering if you could say that this sort of measures the continuity of this function, right? So how like continuous and smooth it is, because it's pretty much the definition of the continuous function that if you vary your coordinates a little bit, then the outcome won't vary drastically.
Yes, that's right. So what you have is an expression for like how like in an abstract sense a function look. And when I've been teaching this before, I've done an exercise where I asked everyone in my class to kind of, okay, everyone draw a function, and then you collect the papers and then you see, okay, so if I just tell you to draw a function and I don't tell you what it should look like, most of you draw things which are like squiggly lines, which are continuous and smooth and all of these things, which is what this covariance structure, as you said, corresponds to, which is these kind of smooth curves. That is a bit of a criticism of this model, is that it does assume very, very smooth curves. And there's some modifications you can do to it to allow for less smooth curves. But, well, my view is that in biology, the data is at a noise level where we will only see these smooth things anyway. Mm-hmm. In physics, it's a bit different.
Yeah, so that's one criticism. As you said, you assume that everything is smooth, but I was thinking about almost the opposite criticism, that this is like too soft a definition, because when I think about spatial variability, just knowing that it's sort of continuous, like I wouldn't call this a spatially variable gene expression, right? So when I hear the expression spatially variable gene expression, I'm thinking more along the lines of, if you go further left or further right or further up, then it will vary in some almost predictable pattern. There will be some pattern. And I believe we can achieve that definition just by choosing a different kernel, right?
I'm not sure if I understand. Are you saying that you're expecting the variance to be spatially dependent as opposed to expression level?
No, I'm saying that, for example, if I had a gene whose expression varies very smoothly, right, then it seems like your method, at least using this standard distance-based kernel, it would detect it as spatially variable, right?
Yes.
Whereas, like intuitively, I wouldn't call it spatial variable because it's still variable in unpredictable ways, right? It's just smoothly or continuously variable.
Right. So I don't see. I think the thing you're describing to me, at least as I'm understanding it sounds quite predictable as opposed to unpredictable.
Well, it is predictable once you have the data. Right. But what I would call predictable is that, for example, if I have the data for the point X, and I could say, okay, if I move like to the left of point X, then I will see like higher gene expression for this gene. Right. But in your model, all you can say is if I go to the left of point X, I will see a similar gene expression, but you cannot say like whether it will go up or down.
No, that's actually not correct. You can extrapolate from this, from Gaussian process models, just as you could from other models. And actually a big benefit with Gaussian process models, for the sake of, for example, forecasting and these things, is that when you do get predictions, it also gives you the uncertainty of the predictions. So you can put a cutoff for how far in the future or how far left you want to predict, given that you accept a certain level of uncertainty in the predictions.
Yeah, that's a very good point, by the way. And I wanted to ask you, because your tool, I think, doesn't actually make use of this kind of uncertainty, right? I think you only use the point estimates.
Well, we don't do any prediction, I guess is the thing. So we only estimate the variance components, which is why we're calling it spatially variable. So there is the larger variance component, which is just the variance of the data as a whole. And then we're decomposing it into two sources. One is just independent variance, and the other source is variance which can be explained by the spatial coordinates. And that's kind of what we're arguing is the spatial variation, something where the variance can be explained by spatial dependence. And then, if I would use this for making a nice, smoother plot, for example, where I do interpolation between the points, I can take my covariance matrix and the data, and then also add new data for the points in between the observed points. And to get these three things together can make predictions of the values between the points where I didn't have any observations. So that's something that you could do, but we're not so interested in that for this study, in particular because there's enough data to just plot the data as it is. Though it is a sensible thing to do a lot of times anyway, because if you want to evaluate these models and how good they fit the data, a good way of evaluating them is by doing prediction and see how good the predictions look compared to the real data. But we haven't done that because we focus the tool to identify the genes. Right. And I would suggest if you want to do more of the actual predictions, should probably look into GPy or a new package called GPFlow, which you can do more of this kind of low-level work with Gaussian processes.
So could you talk about other kernels and what they sort of represent, what they're useful for?
Yeah, so the thing I described before, where just if you have a point and then you let the covariance decrease as a function of distance to that point, we usually write that out as exponential with a square distance in it. And that's usually called the Gaussian kernel or the squared exponential kernel. And it's probably the most common kernel there is, and it kind of makes functions which are infinitely differentiable and really smooth looking. And it's kind of a general kernel. Almost everything can be represented by this kernel. But you can enforce some stronger constraints. So something we're doing just to kind of classify genes is that you can define a kernel that's periodic. And if you have a periodic kernel, you do say that the covariance decreases as the distance from your point, but then it starts increasing again. Because for example, if you have a sine curve, you know that 2 pi away from any point, you should have the same value. So then you will have a period of 2 pi, and you know that no matter where the curve starts, you should get the same values a certain distance away. And this kind of allows you to find things which have periodic structures in tissues, for example. So this would be then a surface that's periodic as opposed to a curve, but that's the idea, that if you can find a period so that if expression value is high in one coordinate, then a certain distance away it will be high again, and then further the same distance away it will be high again. And this is a stronger assumption than the Gaussian kernel. But this stronger assumption means that when genes fit this assumption, it's going to get a better model selection value. And you can find genes which fit this better than the general model.
Right. So I think this is what I was alluding before, that I think this kernel, if gene expression follows this model with periodic kernel or maybe linear kernel, like those actually expose some interesting patterns in gene expression, right? You can say something meaningful about the spatial distribution. I think the difference is that with the Gaussian kernel, if the 2 points are far away, there is no connection. Whereas with the linear or periodic kernels, if the 2 points are far away, but they're related in some way, then there is still connection between them.
Yeah, and there's actually a kind of balance here, and it's something which is sometimes frustrating, which is that sometimes you want to see things which are generally interesting, so you don't want to make assumptions on how things look. And then this Gaussian kernel is really good, so you can find things which are used, like you can explain the data in terms of this, for example, in time. You can predict given the time and your observation data like any other data with it, and it makes really smooth curves in these things. But in many cases, this doesn't help you so much, because now you only know that this gene is here, but there are a lot of follow-up questions like, well, when does it start expressing? Or for how long is it expressing? Or like, how far away is it expressing from another cell, or something like this? And that's kind of, it's both a strength. It's definitely a strength if you're not sure what you're looking for, which is this non-parametric models, which this is a non-parametric surface, basically. Mm-hmm. But then, if you want to know, like, which genes go up as I go laterally across an organism, for example, then just finding genes which are covarying with the locations in the organism is not going to answer that question. Then it's better to move to a parametric model, where you can say these kind of things. And the periodic kernel and the linear kernel are more parametric in this, in particular the linear kernel, because that is equivalent to linear regression in some sense, but it does it by variance decomposition.
Right.
And that usually can tell you things like, as you move across the surface of the tissue, then as you move northwest of something, then the values goes up, which is sometimes interesting. And it is a bit more interpretable, but you have to kind of look at this balance and see, like, okay, do I want to find anything that's doing something interesting? Because that might give me information about some particular period of time or some particular region of the tissue where a given gene is like very concentrated. Or do I want to like actually find a curve that fits, or like a very particular surface that fits the tissue expression?
Got it. And so your tool, SpatialDE, does it it tells you which genes are spatially variable, but does it also tell you for every gene sort of what kernel best fits the gene and with what parameters?
Yes, it does. So it has kind of two key functions. One is a significance test, and this significance test only uses the very general spatial Gaussian kernel. And that's kind of because that is the one that allows us to find the more general patterns. And as I said before, so if you have something like a sine curve, you can fit it both with a periodic kernel and with a general kernel.
Right.
And to keep things kind of comparable, to make the significance test valid, we do the significance test only using the general kernel. But then the genes which are above a significance threshold, we also then fit several other kernels to do kind of a model selection run to see to kind of like break apart, or to kind of move from this general kernel and see, okay, can we like fit something more parametric to these general patterns and see if we can see something there. So at least for now, what we can do is that we can find periodic patterns and strictly increasing patterns using the linear kernel. And then, we're kind of wondering if it's possible to like find other kernels that would find something like interesting, or something that researchers often wants to find in tissues. We haven't thought of anything yet, but like increasing the number of like interpretable kernels would be quite nice.
Mm-hmm. And so in your experiments, what did you find? Like what rough percent of genes are just generally spatially variable versus like periodically or linearly variable?
In most cases, or out of the genes that we do find, which is at least in the 2 spatial transcriptomics datasets that we reported, a very small fraction were significantly variable.
At least in some way, right? Like with the Gaussian kernel.
Yeah.
Mm-hmm.
And then in general, we found a very small number which were linear and a handful of genes which were periodic. That depended a bit on the dataset. So one dataset is a breast cancer dataset, and we found actually the genes that we found which were periodic were kind of interesting because they sort of seem to correspond to invading cell types in the tissue, like immune cells being kind of spread out across the tissue or like peppered across the tissue. The other dataset which I described before is this olfactory bulb from mice. And we did find a bit more periodic genes there. And in many cases, those genes corresponded to being very highly expressed inside, like in the middle of the 2 hemispheres of the brain. So the period is essentially finding the fact that there's symmetry in the brain, like left-right symmetry. So that's what I'm saying before. So if you're in the middle of the left hemisphere and you have a high expression, but it's also finding that the same gene will be highly expressed like 1.1 millimeters away in the right hemisphere. So it's identifying this kind of pattern as periodic, which it is because it's not repeating very often, but it has one very, very clear repetition, which it identifies.
Interesting. So those genes that are not spatially variable, according to the very general Gaussian kernel, how do you think about them? Like, are these genes like an evidence that everything is so chaotic, or everything is so like uniform and homogeneous?
Yeah. So we don't distinguish between those cases. Or chaotic is kind of a strong word, I guess, but what we're identifying here with our test is basically genes which depend on the tissue, like in the global tissue structure. Of course, many things do not depend on this. So, like, and especially if we use the spatial transcriptomics as an example, where each kind of coordinate represents between 10 and 100 cells, you might have some cells there which express some kind of genes and some other cells which do not. But the fact that the cells are there do not depend on the spatial coordinate per se. So it's more small scale structure in the tissue, which would not necessarily depend on the tissue structure itself. There's also a lot of other gene regulation which happens by necessity in the tissue cells, but are not necessarily regulated by the fact that they are in a structure. They might be, for example, cycling or something like this, and that doesn't really depend on the neighborhood as much as other genes do.
Right. Could you talk a bit about normalization on the cell size that you did?
Oh, yeah, of course. So for the spatial transcriptomics, we kind of deal with it as we do for RNA sequencing in general. And something that you generally have to do is that you have to account for the fact that different samples have different sequencing depths. So we do the same kind of normalization there when we use normalize to the total number of counts.
Right.
For the other data sets, we we were kind of surprised about this, but we did find that we had to do the same kind of normalization. And luckily we could use the same normalization, which makes it pretty general. But we found that we have to do it for a different reason, which is, so if you analyze so I said we have these 2 data sets, one from seekfish and one from merfish. If you do, or the data that I could get from seekfish was just a table basically that had, okay, this is a cell which has this coordinate and this expression value for this gene. For the merfish data, they actually provided us, so Jeffrey Moffitt, who is the author, I communicated with him a lot to understand the data sets, and he explained that actually they have you get the coordinate of each transcript, and it's and like a membership of a cell, so you get it on a subcellular resolution.
Interesting.
And this way you can also get, because the way they have saved all the data, you could also get the area for the cell. And something we noticed, we saw that there was a dependence, like on the total number of probes that we saw for each cell and any given gene. So what that means is that if you say, what's the expression of BSN? And I could ask, what's the total number of counts in the cell? And then you respond with, then I can predict what the expression of BSN is. That feels like it's not very biological, or like not very interesting. And it turned out if you looked at the area also predicted the expression of each gene. So, or that there was a very strong correlation between the area and each gene's expression. So it seems that the area of the cell will determine the expression of every gene. And this is actually, there's a really cool paper from Arjun Raj's lab recently where they did some experiments fusing cells, and they saw that if you increase the volume of a cell, it is going to respond by increasing the expression of all genes. So there's kind of an inherent relation between the size of a cell in terms of volume and how many transcripts the cell produces of a given gene. And of course, this is interesting, and the sizes of cells is an interesting biological thing, but we thought to determine interesting gene-specific spatial dependency, we don't want to look at this effect alone. We do a processing where we also look for how much the size of the cell depends on the spatial coordinates, and this tends to get a pretty high pretty good p-value as well. So in many cases, the sizes of cells or the total number of counts of cells is also spatially variable. But it's just one aspect, and further from that, we look at how much of the spatial variance is explained after we have accounted for the fact that cells have different sizes.
So you said something interesting that, like, you have coordinates for individual transcripts, right? But then you have to assign them to the cells. So do you have also the some kind of coordinates of the boundary of the cell, or how do you do that?
Yeah, I mean, so this is data that was that was made public by the different groups, or in this case, Shuai Zhang's group. From what I've been trying to look at their software packages and stuff to try to do this kind of thing myself, to do the segmentation of the images into the cells, and it seems quite difficult, to be honest. And I'm glad they published the data in such a way that I did not need to do that. But there is this kind of image analysis where you try to identify where the edges between cells are, and then you make a mask around it and say, okay, within this little polygon, there's one cell, and every transcript that we identified within that cell is then assigned to that cell.
Right.
So that's kind of how the counting happens in the single molecule fish world, the in situ hybridization world, is that you just kind of define what a region that you call a cell, and then you just count in the image how many spots you see inside the cell, and that's where you get the count. Mm-hmm. So it's way much more direct than, for example, RNA sequencing. And that's kind of so a single molecule fish is sort of the gold standard for transcription to quantify the number of transcripts in cells.
Got it. So now that you're moving labs, what is the future fate of SpatialDE? Like, will you continue to maintain it, or will someone in your old lab will continue to maintain it?
I'm going to continue to maintain it for a while. We have some follow-up things that we're planning to include in it, and we're also starting to work on an R version. And
so the current version is?
It's in Python.
In Python. Mm-hmm.
Yes.
Do you want to re-implement everything in R, or just provide an R interface to the Python code?
I would prefer to implement everything in R. The experience I've had with these different packages that allow you to bridge between them haven't been optimal, so I would probably work on implement it completely in R. But we would like to add the extra functionality that we're working on now, like the future work before. So there's some ideas on how to take this further and answer more questions using the same kind of data. That's ongoing work.
So the goal of converting to R, is it to like provide a better interface for our users, or you think that it will actually benefit the software and it will continue to be like a command line program, but it will be better when written in R?
Oh yeah, that's true. I don't really use the command line version so much myself. I use it more interactively, and then I make scripts that runs it. But yeah, that's true. I actually forgot that I made a command line version.
Oh, interesting. I sort of assumed that it should have a command line version.
Well, so it's because it's so fast, it's pretty nice to just run it with your data, and then you can, I want to try with a different parameter, then you run it again. So that's usually how I like the same way you would use something like DE-seek in R.
Mm-hmm.
But I haven't thought about what we would do with the command line, but maybe we'll keep that. But it's not for speed or so, it's more for the community. So the bioinformatics community is very R focused, and I want people to use it, and it's much easier to get people to use R packages than to use Python packages.
Mm-hmm. Cool.
And hopefully there's been some developments in the single cell field in general, or where they've made really good data structures for these things in R, and I would like to make use of those rather than my more ad hoc solution here.
And so in your new job, if you don't mind sharing, will you continue this line of research, or are you switching to some new areas?
We'll probably continue to some degree. It's still a few months before I move, and I haven't finalized it. But yeah, I think will continue with somewhat similar things.
Cool, got it. Okay, we'll be wrapping up. Is there anything else you would like to talk about that we haven't covered yet?
No, I think it's fine.
Cool. Well, Valentine, it was a great pleasure to talk to you.
Oh yeah, same to you. It was fun.